Hi everyone. So if we could get the video rolling, we'll get started. That's right. How's everyone doing tonight? Can we turn the volume up a little bit? A little bit more? Just met a girl named Jenny, and suddenly that name will never be the same to me. The Jenny just found me wrong, to Jenny. They found how wonderful love can be. Just kidding, just kidding. So, I'm not going to torture you too much with my singing. We're going to do a little bit of science and have hopefully a lot of fun. The presentation is called Absolutely Fabulous. MAB is a monoclonal antibody, and Fab is the antigen binding fragment of the antibody. What we're doing right now, this is the opening dance. Open laptop style. Can you turn the volume up some more? That's right. Yeah. All right. Some people will recognize the song, others maybe don't. This is our star, the antigen, aka antigen. So the song goes, antigen, you know that it's true. Antigen, I couldn't exist without you. So this, what we see up here is the Fab right there, and the antigen binding site highlighted, and the antigen fitting into it. This is a simplified diagram. The structure is a lot more complex, but this is basically all you need to know to understand antigen-antibody interaction. So this is K1000. Going to be very funny. Pay attention. Dansai Uno! To quickly explain, we're all dancing. Me, I'm in the middle. I'm a immature MAB, a monoclonal antibody. I haven't undergone affinity maturation yet. The person who just left the screen was a mature monoclonal antibody who has undergone antibody affinity maturation. We're dancing here, showing that we're first starting out a little bit further. We may bind, we may dissociate. Eventually we're trying to get together, but actually it's not going that well, to be honest. You know? When an antibody first starts out, it's not very good. Normally B cell produces IgM, which is pentavalent or hexavalent actually. This helps it bind, despite the poor binding affinity. There's a poor binding strength of the antibody. Never bind anybody but me. Our affinity, but you gotta promise. Promise you won't take, 'cause I want you to hold me. Binding, way past forever. Okay, so I won't do too much singing, I promise. Anyone who you can remove your earbuds. We'll take a short break from singing right now. Girls like that. So this is the mature monoclonal antibody telling me how I need to fix my ways. So you see in the lymph node, you have B cells ready to explode. They're basically ticking time bombs. You have antigen presenting cells presenting antigens. The whole thing is a huge competition. They got to get to the antigen, they have to bind it in order to get signals in order to survive. The best binders, they can phagocytize, pull the antigen right off of the antigen-presenting cell and interact with T cells. Cells that don't do that are destined to die through apoptosis, programmed cell death. Basically what you see here is a fail, right? This cell right here turning into bubbling, it's apoptotic. Now, looking at affinity maturation on a macro scale, you see a lot of different cells. On the x-axis, we have antibody affinity maturation, as the cells are dividing, and then you have cycles of death. Antibody affinity is increasing. That's right. The goal is to have the best binding clones possible. So the product of this is a plasma cell which produces many, many copies of antibodies that can bind the antigen on the surface of a bacteria or some other kind of pathogen. Yeah? So that's affinity maturation shown in a more artistic way. Yeah? Now we're going to hopefully, can we turn the volume up some more?
Absolutely.
Even more? That's right. Is everyone ready? How's everyone doing?
Let's do this.
Okay.
Today we'll be more like this associate class.
I didn't memorize the lyrics.
Get amplified infinitely as long as you're around. You think that anything better when you and I are around. You know we'll fight for a long time. And even if by chance we do great war, must be
I remember the chorus.
Pathogens they fade till the end of time. Our love is forever the line. Pathogens produce sweet substitute. You know what we gotta do. Pray for each other. Rectify together. Girl, wait until you see how I need ACDC.
So right now in this part of the presentation I'm talking about different effector mechanisms that antibodies mediate. So these include complement-dependent cytotoxicity, which is involves serum proteins that cause cell death, and also antibody-dependent cellular cytotoxicity, which primarily involves NK cells secreting various factors which cause targeted cell death. And also antibody-dependent cellular phagocytosis, which involves swallowing the cells by macrophages. All this is thanks to the Fc of the antibody. So basically, antibodies have a range of effector mechanisms that they can employ to remove pathogens from circulation. That's it.
We can keep
There's
getting sick. All we have to do is stick.
That's it.